Let's do a quick review of DNA and the PCR reaction. And when we start with DNA structure, I want to start with the nucleotide. And the basic components are going to be, there will be phosphate groups, and this is showing with one phosphate. There will be a 5-carbon sugar called ribose, and there will be a nitrogen base or a nitrogenous base. Now let's look quickly at those nitrogenous bases. There are 3 different kinds of pyrimidines, and we'll see in DNA cytosine and thymine. In RNA nucleic acids, we'll see uracil substituted for thymine. The other type of nitrogenous base, the other type are called purines. They're larger and they have a this two-ring structure shown here, and you'll see guanine and adenine. All cells have DNA and RNA, and as I pointed out, one way you can tell the difference between DNA and RNA is that RNA will have uracil and DNA will contain thymine in its place. The other way that you can tell is by looking at the sugars. So let's look over here. In RNA, the ribose has 2 OH groups on the number 2 prime, which you can see here. This is the number 2 prime carbon and the 3 prime carbon. In DNA, what we see is deoxyribose. Deoxy means without an oxygen, and that's on the number 2 prime carbon. It has just an H and not an OH. It is important to point out that they both have this 3 prime OH, and that 3 prime OH is very important because a new base, a new nucleotide joining the molecule will be added at that 3 prime position in both DNA and RNA. Let's look at that numbering system in a little bit more detail. Here's GMP because it has a guanine, and the MP stands for monophosphate, meaning mono is 1 and it's only got 1 phosphate. In this case, let's look at the ribose. This is the form that you would find in RNA because both the 2 prime and the 3 prime carbons have an OH group there. Now let's look at that in a DNA molecule. So this is double-stranded DNA, and that's because we've got a strand here. The strand refers to the fact that we've got a covalent backbone. The phosphate sugars are covalently bonded to one another, and they have, of course, the nitrogen base is also attached, but this is another strand over here. Now the 2 strands are like lanes on the highway. One is oriented with the 5 prime end on the top and the 3 prime end on the bottom. The other is oriented just opposite of that. And notice that, of course, when we say it's oriented opposite, everything is flipped. This carbon is with its oxygen at the top, and this carbon is with the oxygen facing down. And notice that the phosphate groups are also flipped in their orientation, because if you think about it like a car driving on the highway, of course the headlights are facing north if you're heading north, and they're going to be facing in this direction on the way back. It's simply the orientation of the molecule. There's another thing I want to point out about this particular molecule. In addition to the backbone, we would like to look at the hydrogen bonds. So notice that we've got hydrogen bonds actually holding those 2 strands together. The pairing is always T joined to A, thymine joined to adenine, a pyrimidine joined to a purine, and A and T have 2 hydrogen bonds between them. That's why we see these 2 bonds here. Guanine and cytosine have 3 bonds. In other words, they're held together more tightly. and so on. So we've got a C G pairing. And it's this hydrogen bonding that allows us to, in a PCR reaction, for example, break these strands apart, because hydrogen bonds are not very tight bonds and heat will drive them apart. This is a more representative depiction of what happens. So again, here's our double-stranded DNA molecule, 5' to 3' up here and 5' to 3' here. When DNA is being synthesized, and this can happen in a cell or in a PCR reaction, the enzyme DNA polymerase, which is shown in gray here, this molecule that's sort of fitting around the DNA, it will join at that 3' end, and that's what we've already been talking about. The reason it can do that particular type of reaction is because in the cell we've also got, or in our PCR tube, we've also got nucleotides. And in this case, we've got TTP, which is thymine triphosphate. That will be able to enter this molecule, and because there's an adenine here, it will base pair with that, because we just told you that A is always going to pair with T. The next joining would be with this C, and we know that it's going to base pair with a G. A guanine is going to come in there. What polymerase does is, notice that this incoming TTP, thymine triphosphate, will move into position here. And what polymerase is going to do is it's going to remove 2 of those phosphates. Those are high energy bonds. This is the chemistry that drives the reaction, and it will join the remaining phosphate, shown right here, and it will join it to the cytosine that was at that 3' end. Actually, it doesn't join it to the cytosine. Let me be more specific. It joins it to the 3' ribose that's connected to that cytosine. So this is a kind of a tight schematic. Go back and look at the previous picture and it will make a little bit more sense. This DNA synthesis or replication, as I said, this occurs in the cell or in a PCR reaction, occurs because we have this piece of DNA up here, which is what we're going to call our primer. And the enzyme DNA polymerase can recognize this piece of DNA because of 2 things. There's a template here. There's a larger strand of DNA that is only single-stranded in one region, and it has a double-stranded piece of DNA, which is the piece here, attached to it, and a 3' OH. So we're going to use that same chemistry to facilitate a PCR reaction. So here's our starting material. We have what's called your favorite gene, and it's flanked by a region of DNA upstream and a region of DNA downstream. And we don't know the sequence of your favorite gene, and this could be, let's call it a 1 kilobase gene. We just don't know the sequence of it. We do know, however, that we've got this sequence right here that's known to us, and we also know this sequence here. So we're going to be able to design primers for those regions. First, we're going to take that DNA, and in a standard PCR reaction, we're going to heat to 95 degrees, and that will denature or separate the double-stranded DNA. So now we have a strand here, and we have a strand here. We have 4 regions, 2 on each strand, where we can make primers. And our primers are just short stretches of DNA that are complementary to a region of DNA that we know. We're going to make a primer for this region. We're going to make a primer for this region. We're going to make a primer for this region, and we're going to make a primer for this region. So now we've got our 4 primers, and we want to stop and make sure that we know what the 5' to 3' orientation is. So I'm going to label each of these. This is 5', the 5' end and the 3' end, the 5' end and the 3' end, and over here this is the 5' end and the 3' end and the 5' end and the 3' end. Now remember, when we are looking at what polymerase will do, because this is an enzymatic reaction that will be carried out in the cell or in the PCR tube, we're going to add at these 3' positions. So that means the next base, in each case, I'm going to put these in a different color, will be added here. So we'll just put an X because we don't know what they are yet, not until it's sequenced. So this strand is going to go in this direction, and this strand is going to go in this direction. And the other strand of DNA, we're going to be adding again to this 3'. So we've got new bases coming in here, and this is going in this direction, and our other primer is leading in this direction. Now, of course, for PCR, what we really want is we want our primer to allow for copying of DNA in the direction so that we're going to cross our your favorite gene. So we want something that's going to move right in here in both cases. So the oligos, the primers that we want to pick, are going to be this one and this one, because in each of those cases they're going to go in the direction that crosses over YFG and gives us information about the DNA in that region. Now, in contrast, these oligos, the one shown in red here and this one shown on the upper strand, they're going to be productive in a PCR reaction, but they won't give us the information that we want about your favorite gene. They're going in the wrong direction.